Good afternoon. Today we are going to discuss about spread plate technique. Yesterday and day before yesterday we have discussed streak plate method, then pour plate method. Today spread plate method. And in the spread plate method, it is a variety of techniques have been developed mainly for the isolation of microorganisms, mainly bacteria, from the specimen or from the sample cultures. And this spread plate technique, it is to plate a liquid sample which contain bacteria so that the bacteria can be easy to count and also to isolate. A successful spread plate will have a countable number of isolated bacterial colonies which are evenly distributed on the plate. So this technique is among the most widely used culture technique for isolating the bacteria, because we can count number of isolated bacterial colonies which are evenly distributed. And in the spread plate technique, this is the overall method of the technique. The picture here, this is the bacterial suspension. There are some similarities with pour plate technique. Look to this picture. This is the bacterial suspension. From this, we are taking 1 ml to a 9 ml diluted 9 ml broth, and the bacterial suspension 1 ml is adding. Now this dilution is 1:10. Total 10, and among them 1 is the bacterial, that is 1:10 dilution. And from this again 1 ml added to the second tube. Now the dilution becomes 1:100. Same 1 ml again adding to the third dilution, 1:1000. From the third dilution to fourth, 1:10,000. From the fourth tube to the fifth tube, again 1 ml is added, and it may result in 1:100,000 dilution. So, and this method in which each dilution we are adding a drop. I think 0.1 ml is better. If we are adding, this is the picture of this one. Diluted bacterial suspension, this picture is same to this picture. We are adding 0.1 ml. If the concentration or the volume is more than the bacteria, we can't obtain any isolated colony. That's why we are taking 0.1 ml. And we are adding 0.1 ml onto this plate. So initial dilution 1:10 is too numerous to count. Then it becomes, the count becomes less, and then we can obtain the isolated colonies. Here we get a well isolated colony in 1:100,000 dilution. Okay, so 0.1 ml of the suspension is added to the method, that is to the nutrient agar plate. Already the agar is present. In the pour plate, what we are doing? We are just adding the bacterial suspension, and above that we are pouring the media. Here already the media is poured and set, and above that media we are adding 0.1 ml of bacterial suspension, and then we are evenly spreading it with a glass rod, which is a hockey stick shaped glass rod. This is that glass rod, and then we are keeping it for incubation. This is the overall method of spread plate technique. Now we can go through the principle, procedure, etc. And this method, same to that method. One more, I am showing this another picture. It is more clarified view. That is 0.1 ml solution is poured onto the solid medium from the bacterial dilution, and we are spreading like this. Spread sample evenly over the surface like this, and then we are keeping it for incubation, and the bacterial colonies which may grow on the surface of the media like this. Now coming to the principle. In this technique, a serially diluted specimen containing 2 or more bacteria or microbe, which can be the mixed culture. 2 or more bacteria. If the bacteria number is more, then it becomes mixed culture. It is used, and it should be serially diluted, and it is spread over the surface of the solidified agar media plates as with a thin layer with the help of a sterile L-shaped glass rod. L-shaped glass rod, using this L-shaped glass rod, we are spreading it evenly on the surface as a thin layer, while the media plate is spun on the turntable. So we can either turn the table while spreading. Then the principle behind this method is that when the media plate is spun or it is turned, at some stage, single cells will be deposited within the bent glass rod onto the surface of the agar medium. So single cells deposited within the bent glass rod or that spreader, which may spread onto the surface of the agar media. And some of the cells present in the specimen or diluted specimen will be separated from each other by a distance sufficient to allow the colonies that develop to be free from each other. That is the purpose of spreading evenly, to separate the colonies which are easy to develop to be free from each other. Next is procedure. Make a dilution series from a sample. First of all, we are preparing the serial dilutions of the bacterial suspension that we are taking. I have shown the picture, that is 1 ml from the bacterial suspension to the 9 ml medium. It becomes 1:10 like that, 1:100, 1:1000, 1:10,000 like that. Pipette out, then from that each bacterial suspension, we are pipetting out 0.1 ml from the appropriate desired dilution series onto the center of the surface of an agar plate. And we are dipping now the L-shaped glass rod into the alcohol. By flaming the glass rod, that is glass spreader which is in a hockey stick shape or L-shaped glass rod, over the Bunsen flame burner. Spread the sample evenly over the surface of the agar using the sterile glass spreader carefully, rotating the petri dish underneath at the same time. Now we incubate the plate at 37 degrees Celsius for 24 hours. Calculating, and after the incubation, we are calculating the CFU, that means colony forming unit values of the sample. Once you count the colonies, multiply by appropriate dilution factor. So after counting the colonies, we should multiply that value with a dilution factor to determine the number of CFU, that is colony forming units per ml in the original samples. CFU per ml in the original samples. So this is the calculation of the method, that is number of the colonies into dilution factor divided by volume of culture plate. So it is dividing with the volume of the culture plate. For example, suppose the plate of 10⁶ dilution yielded a count of colony 130 colonies are present. So how we will calculate the number of the bacteria in 1 ml of original sample can be calculated as 130 into the dilution factor is 10⁶. So it will be 1.3 into 10⁸ or 130 like this. The value will be like that. Now next is the application. The spread plate technique in conjunction with serial dilution. It is a valuable research tool and it helps in demonstrating the cultural characteristics of the bacteria. Helps in determining the cultural characteristics, for example, color of the bacteria colony, texture of the colony, size, elevation, etc. of the colony can be demonstrated. To isolate the bacteria in discrete colonies from the specimen containing more than one bacteria. So we can isolate the bacteria in discrete colonies. And also for determining the sensitivity or resistance of the bacterium towards the particular drugs or antibiotics or test substances. And also to obtain sufficient growth of the bacterium for various biochemical and other tests. And also to estimate viable count of the bacteria in the specimen, to maintain the stock cultures, and to transport or short-term storage of the specimen. We can do, for example, in the case of stab culture. So we can do like that. So now what are the advantages? Heat sensitive microbes are not affected. No subsurface colonies appear in spread plate. So isolation of organism is easy. So no subsurface colony appear in the spread plate. So isolation can be easy because they are present all in the surface. Next, what are the limitations? Strict aerobes are favored while microaerophilic tends to grow slower. Crowding of the colonies, there is a problem of crowding of the colony and it makes the counting of the colony or enumeration difficult. Accurate dilutions using pipettes should be made. Accurate dilutions. So using pipettes we can take the dilution of 0.1 ml etc. That we are using pipettes for taking accurate dilutions. Volume not greater than 0.1 ml can be spread on the nutrient agar plate because it would not soak well and may result in colonies coalesce as they form. So volume not greater than 0.1 ml can be spread. So if we are spreading more than 0.1 ml, what happens? It may result, it would not soak well and it may result in the colonies thickened, heavy colony formation and which may each coalesce as they form. So these are all about spread plate technique. I hope you understand. Thank you.